The principal objectives of the Clean Water Act are to restore and maintain the chemical, physical, and biological integrity of the nation's waters. Thus, the condition of specific waters or water bodies is determined from the combined measures of physical, chemical, and biological characteristics of each type of water body. This presentation provides an overview of the biological criteria that states and Indian tribes are to adopt to meet the objectives of the Clean Water Act. More specifically, I'll be discussing biological criteria as they relate to the Water Quality Standards Program. Although referred to as biological criteria, this is a new approach and may be applied differently than traditional chemical and physical specific water quality criteria. States adopt biological criteria into their water quality standards, which are subject to EPA review and approval under the Clean Water Act. When I refer to states, I'm also referring to Indian tribes, because Indian tribes may qualify for treatment as states in the Water Quality Standards Program. Let's begin with some background. Water quality standards are laws or regulations which consist of the designated use or uses of a water body or segment of a water body, and the water quality criteria necessary to protect the designated use or uses of that water body. Examples of uses are public water supplies, propagation of fish and wildlife, recreational purposes, agriculture, industrial uses, navigation, and other such uses. Criteria are limits or carefully defined guidelines on a particular pollutant or on the conditions of a water body that are designed to protect and support a use. When criteria are met, water quality is at a level to protect designated uses. Water quality standards also contain an anti-degradation policy and a method for implementing it. The anti-degradation policy sets minimum requirements which conserve, maintain, and protect existing uses and water quality. As I mentioned, the objective of the Clean Water Act is to preserve the chemical, physical, and biological integrity of the nation's waters. Chemical and physical characteristics have long been used to measure water quality. However, as we learned more about aquatic life, it became apparent that biological evaluations were also of great importance. Biological criteria, or biocriteria as they're usually called, are based on direct measures of the biological integrity of surface waters, and thus they provide a valuable assessment tool for evaluating the integrity of our nation's waters. Biocriteria augment but do not replace the chemical and physical elements of water quality programs. A primary strength of biological criteria is that they detect water quality problems that other methods may miss or underestimate. Procedures for developing biological criteria apply to all water bodies, including rivers, streams, lakes, wetlands, estuaries, and coastal waters. Biocriteria are expressed in terms of narrative statements or as numeric values that describe the biological condition of aquatic communities that live in waters of a given use category. Narrative biocriteria are general statements of attainable conditions, such as fish communities as naturally occur. Such a statement may be further refined by lists of species one expects to find in a particular body of water. In the case of a mountain stream, the list may include trout, sunfish, and minnows. Numeric biocriteria, as the name implies, are specific quantitative indicators of a condition of a water body. Numeric biocriteria may be measures of community structure, such as the number of species in the aquatic community, or they may be measures of community function, such as nutrient cycling or the presence of different feeding mechanisms. For example, filter feeding, leaf shredding, and predation. Now that you know more about what biocriteria are and about the role they play, it's time to talk about how they're developed and implemented. As we consider the development of biocriteria for use in water quality standards, we find that 5 major steps are required. The first step is to apply standard protocols. The second step is to establish reference conditions. The third step is to establish biocriteria. The fourth step is to conduct a site survey. And the fifth step is to analyze data for impact. Now, let's take a closer look, starting with step 1. A protocol would include the detailed instructions and procedures to be used to obtain information on the aquatic life in the water body. This protocol also ensures a consistent method of data collection to provide that information. Basically, to standardize the protocols, each state should validate proposed protocols through pilot studies. These pilot studies help to ensure that the protocols for the biocriteria program will provide reliable measures of the biological condition of the surface waters of the state, test for impacts on water bodies, and determine any impairment of water bodies. To assure that the results are scientifically sound, it is essential that each step employs reliable, standardized methods for measuring and comparing the biological status and integrity of any given water body. The next step is to establish reference conditions. This is an important step because establishing reference conditions helps to set the biological condition that can be expected for the water body that's to be evaluated. In most cases, the reference condition will be based on a site or sites that are the least impacted or disturbed, and that is the closest to a pristine condition that can be found within that ecological region. If one is to evaluate a small stream, the reference site should be another stream of the same size and with similar characteristics. Similarly, if one is to evaluate a lake, the reference site should be a lake of comparable geographic origin and in a natural or minimally developed condition. Thus, the reference site establishes the unimpaired baseline for comparison with the site to be evaluated, which is often referred to as the subject water body. The methods that are used to characterize the reference condition will be the foundation of the biocriteria that are to be established, and they'll be used to evaluate the subject water body for the purpose of making a determination of whether the attainment of a designated use has been achieved. By designated use, I mean the type or quality of the fishery or aquatic life use to be protected. Examples of 2 designated uses are a cold water stream that supports trout, or a warm water lake that supports bass. Now, with the standard protocols developed and reference sites established, the next step is to establish the biocriteria. Biocriteria are established by applying the standard protocols to the appropriate reference sites for each type of aquatic life use designation and water body. The results of the biological surveys of the reference sites are evaluated and are used to set quantifiable measures of the reference conditions. They are the standard by which the subject water body is evaluated. These measures of the reference conditions constitute the primary elements of the biocriteria, but are not the only basis for the biocriteria for the type of use designated for the subject water body. States may decide to improve the subject water body beyond the present condition of the reference site. So, after the biocriteria have been set, the state or Indian tribe conducts a site survey of the subject water body for subsequent comparison with the appropriate biocriteria that have been established in Step 3. To do this, a habitat evaluation must also be conducted to determine whether the physical environment at the subject water body sampling site is comparable to that of the reference site from which the biocriteria were derived. Assuming that the habitats match, a biological site survey is conducted to determine whether the biological integrity of the subject water body is consistent with the biocriteria. Keep in mind that the survey of the subject site must use the same biosurvey procedures and protocols that were used at the reference sites. The final step in developing biocriteria is to analyze data for an impact to the subject water body by comparing the data from the biological survey of the subject site with the established biocriteria. This final step requires the use of appropriate statistical or modeling approaches to determine the impairment of the subject water body in terms of the attainment or non-attainment of the designated aquatic life use. So, to summarize to this point, these are the 5 basic steps in developing biocriteria as a part of a state's water quality standards program. To support the quality and reliability of the biocriteria process, we must also take into consideration 3 very important activities. The first is the selection and evaluation of reference sites to support development of biological criteria. The second is the proper measurement of the structure and function of the aquatic community at reference and subject sites of comparable habitats. And the third is the analysis of the results of the biological survey of the subject water body to determine impact and to determine attainment or non-attainment of a designated use. Let's explore them in more detail one at a time. In selecting and evaluating reference sites, keep in mind that reference conditions should embody the characteristics of water body segments that are the least impaired by human activities and should represent the attainable biological conditions required by the aquatic use the state or Indian tribe wishes to protect. There are 2 main approaches to establishing a reference condition. One is to use site-specific reference sites within the same water body. This is the case, for example, when an upstream site is used as a reference for a downstream subject location, or when a nearby undeveloped coastal area is compared to a similar subject site. Site-specific references are useful in controversial situations. However, the site-specific reference approach may be too expensive if used routinely for every site evaluated, and it may prove to be unsatisfactory with multiple discharges, such as sewer pipes, and non-point sources, such as runoff from large land areas. Therefore, EPA also recommends the adoption of a second approach, the use of a regional reference site or sites. These ecologically similar regional references form an excellent basis for comparison of many water bodies. As with site-specific references, the process of selecting and evaluating regional references must be well planned to meet scientific requirements, to maximize information, and to minimize cost. The U.S. EPA is currently helping states in these efforts. EPA consults with states and assists each state as it designs regional or eco-regional databases to fit the state's particular needs. Of course, the effective characterization of the biological reference condition must include the actual measurement of the structure and function of the selected aquatic community. The design must incorporate data collection protocols that ensure the optimal characterization of the component that best represents the state's surface waters. In biological surveys, data collection protocols should incorporate the spatial and temporal scales that are responsible for much of the natural variability of aquatic systems. For example, samples taken before and after the seasonal emergence of aquatic insects cannot be directly compared. Many measures of the community structure and function can be used to describe the components of choice. An evaluation of the number of species is the simplest. However, EPA recommends a multiple measure approach. Examples include the rapid bioassessment protocols developed by EPA. Integrated measures such as these can be used to help set the actual biocriteria for incorporation into state water quality standards. The third activity is the analysis of the results. Proper analysis of the findings is as important as using the appropriate evaluation methods and techniques. A complete discussion of the analysis process is beyond the scope of this presentation. Nevertheless, let me say again that use of biocriteria in water quality standards requires careful application of established scientific principles, methods, and statistical tools. When impairment of the designated use is found, the next step is to make a diagnosis of the probable cause. Obvious causes, such as point source discharges, are generally readily identifiable. But keep in mind that there may be other sources contributing to the impairment, such as non-point sources. In cases where no obvious causes are observed, the diagnostic procedure becomes a repeated process of investigating and testing until the causes are determined. Now, let's review. The development of biocriteria adds another dimension to physical and chemical criteria for a more integrated assessment of water quality. Biological assessments, when used in conjunction with traditional methods, will give a better assessment of the overall ecological integrity of the subject water. Narrative and numeric biological criteria are statements of the condition of aquatic biota that are attainable in order to meet a designated use or uses. A reference condition must be selected that is representative of the least disturbed water bodies for each designated use. Biological surveys of subject water bodies and of reference water bodies must be conducted in a standardized fashion so that ecologically relevant and statistically rigorous conclusions can be drawn. A finding of water body impairment based on biocriteria must be confirmed by testing whether the habitats of the reference and subject sites are indeed the same. And when findings and analyses show that a designated use has not been attained, a process of diagnosis of the cause of the impairment is to be undertaken. Biocriteria are an important addition to a state's water quality management program. This will become increasingly apparent as more states incorporate biocriteria into their water quality standards. The EPA develops technical guidance and other information to assist the states in meeting the requirements of the Water Quality Standards Program. These informational materials are augmented by technical assistance workshops and individual consultations. Additional information about the Water Quality Standards Program may be obtained from these addresses and from the following EPA locations: